jumping lab. So,
All right, I'll share my objective. I'm not thinking it's correct or anything. I just said to determine if it is possible to predict how high a person could jump if you know their BMI, calf size, circumference, and height.
I mean, that would work. I would say in general, you kind of wanted to have a question rather than a statement like that.
Okay, right.
How
Yes,
does the independent variable affect the dependent variable? So what you said isn't wrong, but in general, as a habit, I would say it's better to get into the habit of just asking it as a simple question about how do the independent variables affect the dependent variables.
So would it be incorrect to say, I kept it super simple, can taller people jump higher than shorter people?
No, that's in
What is
fact, so
height?
that's, and one thing is it's not using all the variables. What are our,
I think mine probably covers it. Mine is, does calf circumference, height, and or weight have an effect on how high someone can jump?
right, so did everyone hear what Sarah said there? Like that in a nutshell is kind of about as clean and mean as you can make the objective, right? It's bringing in the specific variables we're measuring, and it's just asking a simple question. Like, how does somebody's height, weight, and or calf circumference, affect their ability to vertically jump? Beyond that, there's, you don't need anything else. Anything else would just be kind of fluff, right? And it's a nice little question. In general, when you are getting to write these objectives, which you're going to be doing fairly often, if you just figure out what are the independent variables, in this case, it's going to be their height, their weight, their calf circumference. There's a dependent variable. In this case, it's going to be the jump height. And then it's just going to be a simple question. How do the independent variables affect the dependent variable? And you're done.
Would it be okay with you if we just as a good practice listed our independent and dependent variables just kind of for our own, like,
Yeah, I think that's probably a good idea. That's
Okay,
a nice
cool.
way to kind of keep track of things. So the objective, a question?
Yeah, I had a question. So once you're, or I can wait when we get to the hypothesis part. But do you cover all three of those variables in your hypothesis, or are you making it on one part of it?
So in your hypothesis, you could just focus on one if you think one in particular is going to be the most important for this lab.
Okay, thank you.
So yeah, let's talk about what are some people's hypotheses.
I can read mine. I said, I believe that taller individuals will be able to vertically jump higher than shorter individuals because their longer limbs will give them a mechanical advantage. And I don't actually really believe that, but it was kind of like, I was like, what can I say that?
And so in mechanical advantage, can you be a little more explicit about what that means?
Just like the longer the lever, getting more.
Yeah.
Oh, now I'm getting into like physics and I don't have the words here, but like.
So the physics, so the angular momentum could have something to do with it.
Okay.
Yeah.
Should I explicitly put that in there too?
Probably, or because sometimes longer limbs could just be you have bigger muscles. If you have a longer leg, you're going to have more massive muscles. So what is it about the longer limbs that you think? That's what, when you have the why, you want to, again, it doesn't have to be right. But it should be compelling. It should at least make sense. You can't just pull something out of your butt and say like, oh yeah, I think because of whatever, it's got to make some sense. But yeah, so longer limbs, that's a good one. What's another possible?
I predict
Go ahead.
I predict that a person with a lower height to weight ratio will jump higher than those with a higher height to weight ratio because they have less gravity to fight against.
So there you're going to probably want to be a little more explicit because height to weight. So if you're going to use height to weight ratio, you probably want to specifically say BMI.
Mhm.
So BMI
Okay.
is going to be their weight in kilograms over their height in meters, quantity squared. Right, that's BMI. And I would say the thing about BMI, the way people usually think about it, they usually think about it more in terms of like kind of category rather than as a continuous variable. Like if you are between 18 and 24.9 or whatever, that's considered healthy. Your height to weight ratio is considered kind of normal. If you're above 25, you're considered overweight. If you're considered over 30, it's considered obese. If you're down below here, you're like too thin. So if I was using BMI, I think it would probably be better to think about it in terms of the category, like having your hypothesis constructed more around healthy BMI versus unhealthy BMI.
I have a hypothesis that uses the normal range. I said, I have a couple. I think that people with normal BMI of 18 to 25 with larger calves will jump the highest. And I think that the higher the BMI out of the normal range, the lower the jump.
Okay, and so both of those sound good. And then the second thing you need though is the why. So what is, do you have a more complete kind of why?
I guess I can put because of the effect of gravity.
So
I don't know.
wanna
I don't
be
know how to
more-
word
Sarah,
that.
I
haven't
actually have
thought
the
about it.
same, Sarah, I have the same thing as you do, but I said, I think this is due to an increase in muscle mass as indicated by the larger calf circumference.
Mm.
And that an average body mass should enable the subject to move their weight with less trouble than those in the overweight category.
Right, and so a couple of things. So if we're looking at calf circumference,
Mhm.
right? So calf is if this is like cut somebody's leg off and just look, here's their bone, here's their gastrocnemius and their tibial, their like tibialis anterior or whatever. You know, how much muscle in here and how much of this is fat is different from person to person.
Right.
Right. So we could say what Ashley is saying, like calf circumference is likely meaning a larger calf means probably more of this muscle.
Mhm.
But again, what else might a larger calf mean?
Might
Well,
mean
that's
more
why
fat.
I said people in the normal range of
So right. So that is
Would
why
have
in your
the highest.
formally written hypothesis,
Mhm.
rather than just implying that, you want to say it explicitly. So when I read it, I know that
Right.
that's what you're thinking. So what would be a way to just say that in a clear way?
Well, you could mention muscle fiber type because obviously fast twitch muscle fibers are bigger than small twitch.
Although we're not really gonna, I mean, I think we're not measuring anything like that. And I think that's getting a little too deep here. So back to Sarah's thing, how would we say state a, because you're the first part of her hypothesis, it's a good hypothesis, but you still need to have a kind of clearly stated like justification of why that makes sense to you.
God, I just don't know what people know. Because they're the muscles in their calf, or the circumference in their calf will be due to muscle mass versus fat mass. No,
So, but
I don't know, I'd
say-
have to
say
think
the
about
whole
it for a
sen- say
while.
the whole sentence.
I would say people with normal BMI of 18 to 25 with large calves will jump the highest due to their calves being large due to muscle versus fat, and muscle being a power source.
A
Yes.
higher ratio of muscle to fat.
Yeah,
Right, so something, I mean, this is where it's useful to try as much
Getting
as
wordy.
possible, kind of, and this process is good to be able to kind of really clarify your thoughts, like, for people in, people in a healthy BMI range, a larger calf is, more likely to be due to like more muscle, and more muscle will generate more force against gravity. Whereas in an unhealthy BMI, it's, more likely that a larger calf is going to be due to fat rather than muscle. So you're not going to expect that relationship to exist for that group, something like that. But you do want to try to make, be clear as much as possible. So if somebody is reading it, they don't have to assume anything about what you were thinking, that you're telling them what your reasoning is. Right? If you don't say it explicitly, there can be a lot of things that you might be thinking. I think I know what you were thinking, but I don't know for sure. So it's important when you do your hypotheses that so I can use this word. I'm sure you've heard it before, but I'll say sometimes this process can feel like you might think I am getting kind of pedantic, caught up in stupid details that are driving you crazy. But for the purposes of this class in particular, stating your objectives, stating your hypotheses is actually a really critical part of this process. In fact, I'm almost tempted to show you an email I just got yesterday. We're asking MAPS, Multidisciplinary Association for Psychedelic Studies. I asked them to share some data from one of their studies that we can use to integrate into one of our studies. And I got a thing back from their research coordinator, says this all looks good. However, I need a little more, exactly what I'm saying here, wanting a little more detail exactly about how this hypothesis works and what you expect, and want to avoid, spurious correlations, blah, blah, blah. So I'm not just kind of making this up. This is the kind of thing that is just part of doing science well, is being able to very clearly state what you're looking for and what you expect and why. So if I am seeming pedantic, it's because partly it's a class where we are learning to kind of develop these muscles to be really clear and concise about what we mean and what we say, and making sure that the person reading it doesn't have to like make any assumptions about what they think you're thinking.
I feel like there should be language courses in science degrees. It's a hard skill to develop.
I was just thinking that, like, I don't, yeah.
Tell the council, no, we got into such a, with counseling around trying to have, we wanted to add that in more, and it's kind of barriers to access and things like that. So there's lots of reasons why they don't want us to do that.
What about just normal medical terminology 1 and 2? Doesn't that kind of do that?
That would be a different thing that you wouldn't need for, that's more, this is more about organizing your thoughts and communicating them clearly rather than knowing the terminology. So but I mean, and that's something you're going to be working on. This is the first one we're doing. This is the first hypothesis and objective. So You know,
So in our lab notebook, if I did my pre-lab, oh, you can't see.
it's supposed to be iterative process. Most people don't. In fact, you're going to find over the course of the semester, you're going to write your objective or your hypothesis, and I'll say, oh, that's a good start, but, and then you'll write it again. It's like, oh, that's better, but, you know, and eventually it's going to be, so that kind of thing is going to be, that's in all of science too. Again, it happens to me when I write things, I get feedback saying that looks good, but, and it's like, oh yeah, no, it's good having another pair of eyes to help you hone your message.
So then here's
So
a
do we just
question
have a-
regarding the lab notebooks. Is this something that once we complete our first couple of labs, will we hand this to you and get feedback on how we've done so that we can kind of change and amend our processes going forward?
Yeah, so normally I'd be there, like normally before we would go on and start actually gathering data for the jumping lab, I would be going around to everybody's lab book and making sure it looked good and giving them a stamp. I always have a stamp for every lab and stuff like that. So when we were fully online, I had people taking PDF photographs of their lab book and submitting it through Canvas. And then giving them feedback through Canvas. But since we're in this weird like nether zone of like, we're kind of in online, but only until like tomorrow, it's like, all right, let's just go into normal mode starting on Tuesday, and I'll give you feedback once we're in person. And again, if you want feedback on anything, I'm happy to look at it during office hours today as well.
And so if we wrote a hypothesis in our pre-lab, like I wrote one here, and then if I'm editing it or writing new ones, do I just keep adding hypotheses? Like
there's different ways you can do it. You could, if you have like your hypothesis and la 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 la, you might be able to change it a bit, like, you know, and la la la, or da, da, da. It could be you can alter it if there's enough room. You can do it in a way that keeps it legible, doesn't make it into a total rat's nest of little arrows and scribbles. Or you might just put a cross here and then a little further down, write revised hypothesis. So it depends how radically you need to alter it. If it's just kind of small edits, you might very likely just be able to do it by amending the original hypothesis. Or if it's more of a radical rewrite, just put a cross through it and do that.
I had a quick question. Just for simplicity's sake, it's okay to make the hypothesis super simple so you can make sure you're just, I don't know, because there's like obviously a lot of independent variables. I literally just said like, I predict people who weigh less will jump higher because there's a less force of gravity. I mean, is that sufficient, or do you want it to be more in depth?
Yeah, no, that would be okay. That would be okay for an initial thing.
And then, I mean, I'm assuming like, because I mean, I've read a lot of scientific journals just from previous classes, but I'm sure you can say like in your discussion, I didn't measure for this, but this is what I saw that interacted with this.
Yeah, so if you look ahead even into, if you have the lab book on you, the more extended analysis is going to be looking at all the variables, no matter what your hypothesis was.
Okay.
So that is actually a very common thing. Like you have an initial hypothesis, you're looking at a particular thing, but you're gathering a variety of data, and you explore, you kind of go and see. Often the story that's most interesting is not what you thought was going to be interesting. Like we are not that clever monkeys in terms of being able to understand and predict everything that's going to be interesting out there in the natural world. We go in with our questions and our hypotheses, and then often get totally surprised, like, oh my God, that was actually really important. And I had no idea to even, luckily I got this little data and I looked at it and I can see that this is actually happening here, because who would have thunk? So yeah, when we get to the more involved data analysis, you will be looking at all of the variables in addition to your particular hypothesis. But for today, just stick to your hypothesis in terms of the initial analysis. I'll talk about the initial analysis in a little bit. First, we got to talk about getting the data. Okay, hypotheses. What about sources of variability?
Shoes,
So types of shoes.
surface,
The surface that they're jumping on, for sure.
I'm putting
Form, subject.
agility and age.
So age could be agility, just how they're comfortable they are in their body.
General activity level.
Yeah, somebody who's just in better shape, even in general.
injury,
Injury. Right. And again, as I mentioned last time, some of these things we can control for. We know that, injuries, particularly things that are going to affect their ability to jump, is really going to affect their jump. So we would probably just say, let's not have any subjects with any injuries that are going to affect their jumping, because that's just going to mess up our data. Things like shoes, we can also get that out of the mix and just say everybody's jumping in their socks. That way we don't have to worry about the difference between Birkenstocks versus Air Jordans in terms of the effect on their jump. The floor, even, we can make sure everybody's jumping in the same room on the same linoleum. So some people aren't jumping in a sandbox or whatever. Other ones we're not going to be able to control. Some people are just going to be more agile or have gotten more sleep the night before or this or that. In which case, that's part of the reason why you get a lot of data points and average them. Like the basic idea is there's always going to be some variables. Like, let's say these are like the true jumps, but the reality is this person jumped a little lower today because they didn't sleep so well. And this person jumped a little higher than they would have because they, I don't know, drank a Red Bull right before coming to class. And this person jumping lower because they had a beer before they came to class. And this person is just like found out that they are like just won the lottery and they're just so stoked about everything. So in general, you're going to assume, even if there is like a true pattern, some people are going to be higher or lower due to these little random things that are affecting these sources of variability. But if you have enough data points, you're assuming that the people who are jumping higher than they would have, like true versus lower than they would have jumped true, are all canceling each other out. So the more you get a bunch of subjects, the more you often get like the true signal comes up above the noise, so to speak, if that makes sense. Right, so that's part of the idea. If you look at any one particular person's jump, any of these sources of variability we're talking about might actually be causing a pretty big problem. But if we have a lot of subjects, then we are assuming that all of the sources of variability are hopefully canceling themselves out. People jumping higher because they are just really perky, and people jumping lower because they're really tired, are all kind of canceling each other out. The more likely for that to happen is if you have a whole lot of subjects that you're pulling from. And what are other sources of variability?
Jumping form.
Yeah, the form. And we talked about the form is going to be important. We'll see that again in a few moments when we look at the video where it describes specifically how the jump is to be performed. Because, your body mechanics make a huge difference in how high you jump. Even your like attitude. Right, in sports psychology, they do all this visualization stuff where, if you go in there and you're about to jump and you just feel feel kind of, I'm not very good at sports stuff. And this is embarrassing. People are watching me. It's like, okay, there, I jumped, happy, I jumped, right? Versus like, oh man, I am going to jump and, impress people. And I feel good about my athletic ability and I'm going to show off or whatever. It's going to make a huge difference. We had one of the groups in the past years, we're not going to be doing these independent research projects this semester, but other semesters where we've had people have to design experiments and carry them out. They took the jumping lab and they gave them these different affirmations where they had to, I'm a great jumper. I am feeling strong. I'm going to like jump for the stars. I forget exactly. They basically had this positive visualization, really getting like stoked. And they had the other group doing like, I feel tired. I feel like leaden, la, 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 these real kind of negative self-talk about their ability to jump. And then other people who had a kind of, kind of the control thing. It's like, oh, the weather is sunny today, or, I had yogurt for breakfast with some granola and then I fed the cat, whatever. So, and, they actually found correlations between these kind of positive self-talk and the negative self-talk and how it affected the, their jumps. So, attitude makes a big difference. Not just in jumping, like in a lot of life. If you kind of dig into something thinking you're going to get out the other side, you've probably got a better chance than if you kind of walk in and thinking you've already kind of failed before you start. So we'll talk more about sources of variability as we go, but just from all of this kind of yapping I'm doing, just to kind of give you a sense, if you don't think about it ahead of time, there's a good chance you're going to get all your data and it's garbage. If you did not control for the shoes by making sure people take their shoes off, probably the effects of shoes on their jumping is a bigger effect on anything else that we're considering our independent variable. So if you have sources of variability that are actually big sources of variability that you're not trying to take care of, then it's just going to contaminate your data to make it hard to tell anything useful from the data, even if you've done your experiment. Also, it's going to cause other people to challenge you. Like, well, how do you know it wasn't just the shoes? It's like, it's because we made sure they weren't wearing their shoes. They all jumped in their socks. So there, right? So that's another thing. Like, so you can, defend your results, or, by saying, yeah, no, I actually thought about that and I did my best to make sure that that did not contaminate my data to the best of my ability. So anyway, what time? Maybe raise your hand if you have not finished writing all of your sources of variability and your hypothesis and your objective.
Even if we need an adjustment now.
Yeah, actually, if you let's take 10 minutes to just kind of adjust them. Because I would like people really kind of going into this feeling that they are feeling solid about what they've got there. So let's see, it's 11:33. At 11:43, we will actually start getting the data, but I am going to just sit here and be available if you have questions or clarifications. But let's just take, Hey Siri, 10-minute timer.
10 minutes, counting down.
All right, so when Siri tells me it's time to go, we'll start the rest of the lab. Yeah, and if you're happy with your objective and hypothesis and sources of variability, then just kick back.
So I know we're gonna be finishing the jumping lab today. Sorry, I had to step out of the room really quickly. My cat was making a lot of noises. But you said we're gonna finish the jumping lab today, but then I heard you also saying we were gonna skip something and come back to it.
The jumping lab analysis with Excel.
Okay, so we're gonna finish like the data collection portion and stuff today,
Yes.
and then and then
Kind
the
of
last.
this initial analysis, but we're going to do a much more extensive analysis later.
Okay, so we just basically what you were just saying is we won't complete the entire thing today. Like the whole.
We're gonna complete this first part, the thing that would have happened on Tuesday. And the thing that we were supposed to do today, according to the schedule, we are going to do on the 22nd.
Okay, gotcha. Thank you.
Hi. So I have a question.
Sure.
I guess this kind of circles back to how scientific the language needs to be. I mean, I know that you're saying to be specific and why. And I apologize if this sounds too simple. But if in my hypothesis, I'm stating that I believe people with a height advantage will jump to greater heights than people who lack an advantage of height.
But so even saying height advantage, I don't know what that means.
I guess
Like,
because
I have
it's a starting point, right? So they're higher. It's kind of
Right.
like, you know,
So height advantage, like I'm only 5'6" and I have a huge advantage if I'm trying to sleep in a car compared to somebody who's taller. So saying a height advantage doesn't mean anything.
okay. So when you say specifics, what I would do is give like a bracket of like
Or you
height.
could just say taller people versus shorter.
Okay,
It's
that's
saying
what
the
I
word
started
advan-
with. I thought it was way too basic.
No, not at all.
Okay. And then I don't quite know how to like how
And it's, if you look at the actual procedure,
Mhm.
it's you mark the wall wherever you're at, and then the jump is how much you can get off the ground compared to your.
Okay,
So
okay,
even if you are 7 feet tall, you're still starting with your baseline mark is wherever it is, and it's how high can you jump above that.
so that basically kiboshes my whole hypothesis. So I should take a different perspective.
Right. So it could be taller people can jump higher, but it's not just because they're starting higher. It's something about, again, like longer limbs will have like
Or muscle
larger
mass or
muscles. Yeah, exactly.
okay. All right, thank you.
Sure. All right, Minnie says it's time to get back to lab. All right, so anybody have any points of clarification or anything before we continue on? Again, on Tuesday, I'll be able to kind of go and look over this stuff since we'll actually be, make sure you bring your lab notebook into class on Tuesday as well. You know, we'll be doing lab. So in addition to your Scantron and your notepad or whatever, make sure you bring your lab notebook. Okay. So, not pages, modules. So what we're going to do, normally this lab would be, everybody would be measuring each other and then jumping. But instead, we're just going to show you a brief little video about how the data was gathered, since we're not actually gathering it, but it's important that you can get a sense of where the data came from. This is going to be different than the chemistry lab. So you don't have to write anything down. There's no personal data for you. You are just going to kind of watch to see how we got the data. And then I'll give you a little spreadsheet with some initial results, as if we, from a previous class. So, at this point, you just kind of kick back and enjoy the ride for the next 10 minutes or so. This is under the module for Jumping Lab Initial Data Gathering and Analysis. I just published this Doing the Lab page. So this is now available if you need to go back and look at it for any reason. And it's, if you are going to have to go there, because that's where the initial data is. But it's basically got two little videos that we're going to look right now. Then there's a little spreadsheet with some data, and I will talk about the expectations there in just 10 minutes after we look at the videos. So here we go. Well, that's not a very flattering picture there, but that's life.
Fun socks.
That's not a coincidence, right? Even I talk about it in the video, even if when you know you're gonna take your socks off in public, I at least I always make sure I put on groovy socks. So here we go. All right, so this is the jumping lab, and in this lab we're going to be taking a variety of measurements off of our subjects and then looking for correlations with their jumping height. So one of the measurements we're going to be taking is their height. Again, talking about sources of variability, your height is going to be really different depending if you're wearing platform shoes or you're barefoot. So before you take anybody's height, you gotta make sure they don't got shoes on. Shoes. Another thing, some people, I don't have poofy hair, obviously. Some people have poofier hair. I want to make sure as you're measuring height that you compress down and you're actually getting your actual height and not the height that they're. So here we have meter sticks taped on the wall. I'm gonna stand here straight and let's see.
Okay.
I'm gonna show parallax.
What was that?
Show parallax. Like, you have to make sure it's parallel, over or underestimated.
Oh, yeah. So it's important that it's parallel or perpendicular to the ruler in the wall, because you could measure too high by doing this or too low by doing this. So you want to have it completely perpendicular. And these meter sticks are marked with millimeter tick marks. So in this case, we want to measure to the nearest millimeter. And then you want to record that in your lab notebook. And also don't forget that there are 2 meter sticks taped here. So any person in the lab is definitely going to be over 1 meter tall. And then you reach to the nearest millimeter here. So in this case, it would be 1.658. And then you can convert that over to meters.
And in the actual lab, you would have more than one person take the same measurements. It's because even though it seems like a straightforward thing, there are so many slight variations, the exact angle of the meter stick, or just somebody's approximation of whether it's up or down 1 millimeter, that can actually lead to a different value. So to try to get at the true value, we take multiple measurements and we can then average them and then assume that that average of those different measurements is approaching our true. Right, and I should mention this idea of the sanity check. Like if we were doing this in person, I promise you, there's always some group that measures the height and writes it down as 65.8 centimeters, for the height of their subject. If you what's wrong with that number?
Very small.
The units.
It's really small.
Yeah.
65.8 centimeters, that is about 2 feet, about 2/3 of a meter. So, metric is how we do this class. And so if you don't have a intuition for metric units, that's another thing you want to start developing. I know in Bio 110, they had you think about parts of your body, like, the width of your finger is about a centimeter or so. So when you think about a centimeter, you can think about it, oh, it's a little thing like that. A millimeter is about the width of your nail. So a meter is about 3 feet, about the fish that got away or something, right? You really want to make sure you develop a sense. Like if I told you that I measured, I'm looking here, like Rebecca's height was 2 feet 3 inches, I made a mistake. It's not true, right? But when somebody says, oh, I measured her height and it's 65.8 centimeters, a lot of times when people haven't been raised with metrics, they're just like, okay, I'll write it down. That's her height. So just part of the sanity check, make sure you're thinking about what the actual units look like. Also, just remember, if I have a meter stick that measures in millimeters, it's going to be like 0, 10 millimeters, 20 millimeters. And you want to use the highest precision that you can get on this thing. So don't just say like, oh, it's near 10. Say it's 10.3 if you have the little tick marks there. All right, let's continue. All right, so this next part is measuring the cap circumference. And again, here's another example where we need
Share
to
the video.
bring in this idea of thinking about sources of variability.
We can't see it.
Actually, do the experiment. All right, sorry about that. And I will also say that thing I mentioned about measuring 3 times, it really does. We have 3 different people measuring, the person's height in their group. It's not always the exact same number, even though you might think, oh, sure, maybe theoretically, or maybe someone's just standing a little like more straight and a little more relaxed on one measurement to the next, or again, somebody tilts the ruler a little bit, or it's getting the same measurement again and again isn't necessarily as straightforward as you think. All right, so this next part is measuring the cap circumference. And again, here's another example where we need to bring in this idea of thinking about sources of variability before you actually do the experiment. Right? just like you have right-handed and left-handed people, there are people who are right-legged and left-legged, have a leg that they prefer. And they're gonna probably have more muscles on their leg that they prefer. So the calves are probably not exactly the same. So it actually is gonna be important to like acknowledge that before we start and then come up with some way to deal with that. And the way we'll deal with it today is we'll measure the calf circumference of their dominant leg. That would be the leg, like if you were told, go hop on one foot from this side of the room to the other side of the room, which leg do you hop on? And that is your dominant leg. So when your subject is getting ready for this, have them hop. See which leg they prefer, and then you want to measure that one. Oh, also, I highly recommend if you know you're taking your shoes off for this lab, wear fun socks.
All right. And we're going to measure it 3 times with 3 different people measuring the subject's cap circumference, and then you're going to average that.
Okay. So, yeah, we did not make a video for getting somebody's weight because that's pretty straightforward. You step on a scale and read it. So what is in kilograms? What would be a typical weight? What is a kilogram in what we're used to? What's a kilogram in terms of pounds?
It's a
2.2.
It's about 2.2 pounds, right? So if somebody is, 50 kilograms, that's a pretty typical weight. That's like 100 something. If you wrote somebody was 100 kilograms, they would have to be big. That's like 220 pounds, right? So again, understanding what the units are is going to be important in deciding whether or not you've made a mistake. The other thing I should mention, we'll talk about a little more later as well, but the idea of significant figures. So Sig Figs, I know you've seen in chemistry already. What is the basic rule for measurements in Sig Figs?
the last number is usually a guess. So when you're looking at a meter stick, you can see, oh, it's 10.5 centimeters, and the 0.5 part would be the guess. Or you could go further depending on how many ticks are on the measuring piece.
Right, so Sig Fig, how does this relate to doing math with measurements?
You want to keep your number of Sig Figs consistent.
So the basic, you're only entitled to as many significant figures as your least accurate piece of data, because that's going to limit the result of your calculation. So if I did some measurements, I don't know, if I was measuring people's calves or something, right? 38.3 and 38.5 and 38.6. So let's say these were my measurements. And I wanted to get the average. And so I said, okay, well, I'm going to do the average, and I'm going to do it fast. Here's my calculator. I'm going to type in 38.3, 38.5 plus 38.6 plus, and then 3 divided by. And this average is 38.46, 6. And I write this down, someone's going to say, that's ridiculous. Now, how can we know the answer out to the 1,000th decimal place here if our least accurate piece of data was only here to the 10th place? And what Rebecca was saying, this actually means 38.3 plus or minus 0.05. It means that, right, so all of this, it looks good. It's like, wow, I know their cap circumference to so many decimal places, but it's meaningless. It doesn't mean anything. So if our measurements have 3 significant figures, you're only entitled to 3 significant figures. So this would be rounded to, this would be 38.5. And that's what I would write as my average calf. So that's important when you're doing calculations with these measurements, which have a limited accuracy like that, then you need to make sure you only include as many Sig Figs as your least accurate piece of data. So, all right, let's continue. All right, so the next part of the little video is just showing you how we actually gather the jumping data. So you can see how we gathered the body measurements. The next part is the jumping data, which is just basically having somebody stand in front of the big piece of butcher paper and jump and make marks. But here you should take a look and see, you can watch me jump. Here we go. All right, so we've measured my height, my calf circumference multiple times, so we get hopefully as true a value as possible. We've also measured my weight standing on a scale. You all know how to do that. And now we're going to talk about our dependent variable in this whole experiment, the actual vertical jump height. A few things before we start that. Another source of variability is the actual mechanics of the jump itself. If you just kind of like that or whatever, it's going to make a big difference on how high you jump. And because we want to look across many subjects, we want to have a standard jump where everybody's doing the same mechanics, which I'll briefly show you. It's basically described in your lab manual. You're standing vertically, your hands by your side, bend your knees, and you're going to extend your knees as you swing your arms up. And you want to have your legs and your arms extending simultaneously so they both are at full extension at the same time. So there's this really nice kind of synergy that happens. And you also want to make sure you're using both arms. A lot of people have this tendency to like jump like that and just throw up one arm and they have half their body is just kind of, you know. So you want to have body straight, bending at your knees, and then in one smooth motion, explode up with your arms going up, your legs straightening, and actually show how it's done. I'm going to have a pencil here where I'm actually going to be able to mark. Now, obviously, we want to see how high can I jump from my normal height. I don't care if a person is taller or shorter. You know, they're going to be able to mark the paper higher or lower depending on their height. That has nothing to do with the jump. So what I care about is where do they mark it if they're just standing on the ground? And then I'm going to be able to jump and mark it at the peak of my jump. And then I'll see what's that relative distance. And so I'll do that. This is my baseline. Put it. Thanks. Ready? And I want to, again, every time you jump, you're obviously not going to jump exactly the same height. So another thing we're going to do, just like we do multiple measurements for height and all that, we do multiple measurements for the jump. So I'll go like that. And holding the pencil with the exact same angle that I'm going to mark at the top of my jump. Get ready. Straight body, fat legs. Go up. There's the second jump. And I'll get ready. And one last time. And my lab partner, if I go in like this, my lab partner's got to say like, "Whoa, that wasn't a. It was nice to watch you jump. But that wasn't a jump we can count because you didn't have to talk before." So as a person, making sure the experiment is done correctly, you need that. You don't want to throw away data just because it isn't what you thought it should be, but you don't want to take data if they actually didn't do the procedure properly. That doesn't. Okay, my third jump. And now I have 3 little marks up there. And now it's time to measure how high my vertical jump is, so we can see how it correlates with my height, my weight, my cap circumference, and some other things.
So next, it's time to measure David's jumps here. So carefully, without falling off, you want to get up here, use the metric system of any ruler that you have. And then, of course, it's not very handy for me to record this. I'm going to have to yell out the numbers for my lab partner to write down in the lab notebook. So I'm going to go from the baseline to the first mark here at the highest point. And it's going to be 47 centimeters and a half a centimeter. So 47.5 centimeters. 47.5. All right, second jump. 46.2 centimeters. 46.2. And the third is 47.0 centimeters. So those are the 3 jumps that they did. All right.
All right. So. La 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 la. Right, hold on. Okay, so there you have seen how the data was gathered, and also talking a little bit about, the reason we do this jumping lab is not because there's some compelling question about human physiology we are solving with this lab. It's more about demonstrating a lot of the things you need to think about when you're doing experiments. Right, so this is, the actual like jumping versus body parts is not the point. That's obviously it's what we're going to be looking at, but things to think about as you think about this lab is also this idea of sources of variability, the little pitfalls about just taking measurements, about how you approach trying to get solid data. So that's part of the reason why, you've spent the last 10, 15 minutes like watching us do this rather than just me throwing the data at you. So it's useful to know where your data comes from, even if you didn't gather it. It's even when you're reading reports about other people's work and looking at their conclusions, part of becoming a more sophisticated consumer of information, particularly scientific information, is, all right, how did they do this experiment? Look at their methods. It's worth looking at their methods and seeing how they did the experiment and see, do you trust this? Just because something gets like out there doesn't mean it's necessarily good science. It's up to you to decide whether you respect this experiment. And then, the more you kind of get under the hood and how this is done, the more you'll have that kind of skill. Okay. So once we do this, usually we have everybody get their data and put it into a spreadsheet. So here is the data. So this is the data that you all have access to. This would be like if we just got one class worth of data. Like I mentioned, it's a lot better having more subjects. For the analysis that we're going to do in a few weeks, we're going to have many classes of data. We're going to have like almost 100 students rather than just like 20, 25 students, 26. But what I would like people to do is just kind of an initial analysis. So the point, what I want you to do with this data is not some really big, fancy, huge write-up, because we're going to wait for the additional data for that. But I would like you to do some initial analysis, trying to decide whether or not this data supports your hypothesis or not. So let me talk about that in more detail then. So this is all on that Canvas webpage. You can just pull this up, it's easy. So let's talk about, so again, we've got our objective, our hypothesis, our sources of variability. We also had our methods, that has to be in there as well. We also have, this is kind of like the data, which is kind of the raw data. But now what's the next step? As we start our kind of discussion, what do we need to do?
Analyze the data.
And how are we going to start? What are we going to look for? How are we going to analyze?
Trends,
What's that?
trends in the data.
In what particular kind of things?
Well, I think each person will analyze it differently depending on what their hypothesis is.
Exactly. So, right answer, right? This is, you know, where you start here is going to depend on what your hypothesis was. So check if data supports your hypothesis. So I think was it Alex was saying that, you know, word your hypothesis was too simple. It was just like weight versus their jump. Was that right?
Yeah, weight versus the jump.
So what would you do if you want to do some initial analysis to see if the data supports your hypothesis or not?
I would just look at the weights and then compare the jumps based on the different weights that were in the table.
But how would you do it? How would you present that? How would you do that in a way that's going to make it clear if there is something going on?
Like if there was a positive, if it's right or if it's not, like just, I don't know, just say like.
Would you make a chart with weight and height of the jump?
Yeah, so if you're comparing two continuous variables like height and jump height, or weight and jump height, then the best thing to do is make a scatter plot, right? So scatter plot, again, I know you've done this in 110 as well. The idea of a scatter plot, typically on your x-axis, the abscissa is typically where you put your independent variable. The y-axis or ordinates typically where you put your dependent variable. And then each individual is going to be a dot on this graph, and then we can look for trend lines. So in fact, what time is it? Yeah, I can do this. I need a plastic ruler or else it's not gonna work. Okay. Since you are actually doing this for kind of actual analysis, it actually is important that you use the graph paper. A lot of times people, when they're first starting out their graphs, they get, in fact, let me start this a little prettier. I'm gonna go, use a ruler, so it's gonna be pretty. people start getting loosey-goosey with their axes. So my dependent variable, this is going to be their jump height in centimeters. Independent is going to be their weight in kilograms. And then what I do is I look at the range of the data, right? It doesn't make sense. Nobody weighs 0, right? If I started my weight at 0, there would be so much dead space on this graph. So I look, I say, the lightest person was about 44. The heaviest person was 105. So I only need my weight to go from like, 40 to 110. So I'm going to do that. I'm going to like, you know, 40, and maybe this will be 50, this will be 60, 70, 80. $90, $100, $110. Right, so I want to make sure I figure out my axis so the range of the data is going to be spread out kind of along the bottom of my graph. It needs to be spread out. I'm actually using this to actually look for trend lines. So you don't want to have everything so squished you can't see what's happening. So you typically want to have kind of, we call the aspect ratio. It's good to have the overall thing kind of have this, kind of like a TV screen kind of ratio, aspect ratio, kind of a little wider than it is tall. And then figure out, I figured from 40 to 110 is the range of my data. So figure out how much each tick mark should be so it spreads out about right. For the jump height, similar. Nobody jumped to 0. So I look at the jumps. The jumps go everywhere from 14 up to a little over 60. So if I go from 10 to 60, I'll probably be okay. So right? So when you are coming up with your axes, be smart about them. Think about what is the range of the data. Actually, I didn't need to start from 5. Duh, that's not what I meant to do. But it doesn't matter. People are pretty low. It doesn't matter. So then I just go person by person. I go, okay, number, I don't know, does anybody have this up on their screen right now? And I can just read it off here. I have,
But you gave pre-con, what is today?
so I have a weight is 60 and the height, jump height is 6, actually weight is 60. And the jump height is actually about 61. So there is that data point. The next person, their weight is 104 and their jump is 27. Weight is 104, jump is 27. There's them. The next person, their weight is 63 and their jump is 40. There you go there. I keep going down. There's weight is 52 and the jump is 28. So you just go like that. Each person, each data point becomes a dot. Each person, their weight and their height become a dot. And then you take a look at what happens. Maybe when I'm done, it looks kind of like this. You know, we are gonna learn how to use Excel to do more formal curve fitting later. But for right now, you can even kind of eyeball it. You know, does there seem to be an overall line that is kind of the average behavior? When you're eyeballing these trend lines, you usually think about where's the line where about half the dots are above the line and half the dots are below the line. Again, when we're using Excel, Excel will actually calculate the linear squares regression, least squares regression. So it could also very well be that you do all of this analyses and the trend line, you know, looks like that. There is nothing. You don't know though until you do it. So if you have two variables that are continuous like this, and I want to see, does there seem to be a relationship? Just make a scatter plot and then draw a trend line to see if there's some relationship, and does that support your hypothesis or not? So does that make sense? So that's one way. And if you have multiple parts in your hypothesis, you know, just if you have one for height, one for weight, one for calf, just pick one. Don't worry about doing it for every single. If you have a multi-part hypothesis, do it for one part. What if I had a hypothesis that healthy BMI people are going to jump better than unhealthy BMI people? What kind of a graph do I use for that?
You would use the BMI against the jump. Right.
Because BMI is healthy and unhealthy, it's more categories. Whenever you're doing things that are in categories, what kind of graph do you use?
Would it be a bar graph?
Bar graph.
We use a bar graph. So what I would basically do, I have healthy BMI, unhealthy BMI, you know, average jump, centimeters. What I would have to do is go to my data. In fact, I'm going to just share that screen again. Go back over here. I would have to look. Okay, this person, healthy BMI, will use their number. This person, nope. So I would basically kind of probably use a highlighter. In fact, let me use a highlighter. I will. So for all the jumps that are healthy, I'm going to pick those. This person unhealthy, healthy BMI, I'm going to pick this one. Unhealthy, 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 healthy BMI again, I'm going to pick that one. This is another healthy, I'm going to pick that one, etc. I can pick all the healthy BMIs, and once I have them, get their average. Add up the number of healthy BMI jumps and find out what's the average number. Add up all of the unhealthy BMI jumps and get an average for that. And then once you have those, you can then put it in here, you know, healthy, unhealthy, average jump in centimeters. Be something like that. So that would tell us, do healthy BMI people tend to jump better than unhealthy? Or if we go to, I think it was Sarah's original one, looking at calf, but only for healthy BMI people. That's totally easy as well, right? All you do is we take these people with healthy BMIs. So it's going to be like, this person is a healthy BMI, this person has a healthy BMI. Sarah, can you mute yourself, please? Healthy BMI, healthy BMI, healthy, 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 unhealthy, healthy, 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 boom, 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 boom. Just for those characters, let's graph their calf versus jump. So I can see calf versus jump, but only for the people with healthy BMI, right? So there's many different ways you can look at this data or answer different questions. We can say just in general, does this independent variable affect this dependent variable, or the jump? Or we can say a scatter plot, but just for the subset of healthy BMI people. Or if we just cared about categories, healthy versus unhealthy BMI, we could just get the average jump for the healthy BMI versus the unhealthy BMI and put them in a bar graph. So does that make sense? So for your initial analysis, I don't want you to go too deep and look. I don't want you to look at all the different variables yet. We are going to do that later. We're going to do it when we have more powerful tools to graph with Excel and all that. For this first step, just look at the data to see if it supports, this initial data supports your hypothesis, and just write your initial conclusion about that. And then we'll be able to look at the larger data set and see if that conclusion is still true when we analyze the data with a larger data point, with the larger data set. That makes sense?
Should we put this table in our lab notebook?
Yeah, you should.
Okay.
Yeah. So any questions about any of this? So for next Tuesday, again, I also want you to have an objective and a hypothesis. This Osmometer Lab. The basic idea in the Osmometer Lab is we're going to have a semi-permeable membrane. And this is going to be sitting in a little beaker of water here. It's distilled H2O. And I'm going to have the same setup. It's distilled H2O. Except in one case, I'm going to have a 40% sucrose solution inside. In the other place, I'm going to have 20% sucrose inside this little tube. Let me kind of add in so what is going to happen if I have a semi-permeable membrane here, and I have sugar water inside, and on the other side is distilled water?
More water is going to go into the 40% solution.
Yeah, exactly. So if I have 40% sucrose on one side, distilled water over here, osmosis. Water is going to move towards where there's more solute, right? That's osmosis. That should feel familiar. The salt sucking in the solute, the water gets drawn towards the solute. What affects the rate? How fast the water moves?
Temperature could be one thing.
Yeah, so temperature does. What is one of the, what's that?
Concentration.
So concentration, but a little deeper. So it's not just the concentration. If I had 40% on both sides, there'd be no movement of water, right? It's not just the concentration, but it's the difference between the two sides of the membrane. It's what we call the concentration gradient. That is something that you need to remember, this word gradient. This is the difference between the sides. What's really going to affect the movement of water across the membrane is not just the concentration, it's the concentration gradient. Like in your, and in fact, if we look at our setup here, our independent variable, the main difference in these two situations is not just that it's different concentrations inside the little tube here. It's that the concentration gradient across the membrane is different, right? This goes from 0 sugar to 20% sugar. This goes from 0 sugar to 40% sugar. So this independent variable is going to be the concentration gradient. People get into trouble here by saying that the variable is concentration, because concentration in and of itself is not going to affect how fast the water moves. You know, our dependent variable that we're going to measure is just the rate of osmosis. How fast the water is moving across the membrane, you know, into this thing. We're going to be able to measure it by seeing how fast the water is going up the tube. You know, which of these setups do you think is going to have a higher rate of osmosis? Let's say A or B.
A.
A, and why?
Steeper concentration gradient.
Yeah, it's got a stronger, a steeper concentration gradient, and the rate of osmosis. In fact, we're going to talk about a variety of other things, but it's a huge thing that affects that rate, is that concentration gradient. So it's pretty simple. The objective is to see how the concentration gradient is affecting the rate of osmosis. Your hypothesis is between these two setups with the 40% and the 20%, which one is going to have the faster or slower, and why? Right, and that's all you need to do in this part. So it should be pretty straightforward. We'll discuss the idea of things that affect the rate of diffusion in much more detail when we get to do the lab next week. But just for Tuesday, I just want you to at least think about what you're getting into and just write in your lab notebook this basic objective and hypothesis for this lab.